Thank you so much, sir. Next, we explore the rapidly advancing field of genetics and genomics and pediatric medicine. It's our great pleasure to have Dr. M.S. Maila Ashfaq, who is a board-certified genetic counselor specializing in cardiovascular genetics and global genetic counseling research. She holds a Master of Science in Human Genetics and Genetic Counseling from Stanford University, California. She is particularly focused on clinical management and counseling of patients with cardiomyopathies and arrhythmias. In recognition of her contributions, Dr. Maila was honored as one of the Dean's Teaching Excellence Awardees of STEAMED Educators Program in 2024 till 2025. I request to please proceed, Dr. Maila.
Dr. Maila, can you hear us?
Yes, I can hear you. Can you hear me?
Yes, we can hear you very well, and we can see your slide as well. Please proceed.
Okay, perfect. Thank you so much. Thank you for the invitation to speaking at this prestigious conference. I am presenting after two excellent presentations. So this is a hard act to follow, but I will try my best and talk about genetics with everyone here today. So let's see. Okay, so my objectives of the talk are that I hope that everyone recognizes indications for genetic disease in children, understand some major types of genetic tests that are available here in the United States, appreciate why genetic diagnoses really matter for children and families, and recognize the role of geneticists and genetic counselors as they practice here. So I'm going to start with the last objective. I myself am a genetic counselor, and I just want to describe to everyone what a genetic counselor is. So I work out of a clinic, and my specialty is cardiomyopathies and arrhythmias, but I also do general genetics clinics out of the Department of Pediatrics. So in our clinics, we have families who come in and we meet with them, we talk to them, we collect medical and family history from the families. And then we think about what are the possible differential diagnoses in genetic disorders that could be reasonable based on the medical and family history. And we discuss genetic testing options with them. We decide on a genetic test, enter the order, send the test, the results come back. Then we discuss the genetic testing results with the families. We talk about what the diagnosis is and the surveillance and management that needs to happen for them. So we help coordinate their care. We assess genetic risk for other members of the family, depending on the inheritance pattern of the condition, and we provide psychosocial support. We also have geneticists in clinic alongside us. So the difference between a genetic counselor and a geneticist is that the geneticist is really involved with the physical exam. They might write scripts for medication if necessary, and they might order other testing as well. So I'm just going to do a little bit of a revision about genetics and DNA so that everyone is on the same page for the rest of the talk. But as everyone here knows very well, that our human genome, which resides inside the nucleus of our cells, contains about 23 pairs of chromosomes in each cell. And the first 22 pairs are known as the autosomes, and they're the same in both males and females. And the last pair are the sex chromosomes, so XX in females and XY in males. A pair of chromosomes are referred to as homologous chromosomes, and we get one member of each pair from mother and the other from our father. And along the length of our chromosomes, if you unravel them and uncoil them, we have our genes. And in humans, there are about 25,000 genes. And this last image just shows us our DNA. So when we think about genes, everyone remembers that there are exons in genes and there are introns in genes. And the exons are the coding part of the gene. So the exons are what gets transcribed into messenger RNA, and the introns are snipped or cut out. They're also in the past referred to as junk DNA. And all of the exons, they join up together, and the mRNA is then translated into a protein. So when we're thinking of changes within genes, often historically, we've been using the word mutation to describe a change in a gene. So now, however, the terminology has changed a little bit, and especially in the clinical environment, we don't use the word mutation to describe a change in a gene. Rather, we say that there's a variant. So when we see a change or a variant in a gene, which is different from the reference sequence. So it's different from what we would otherwise expect. We then go on to classify whether that variant is something that could potentially cause a problem or be pathogenic, or if it's just a benign variant. Because me and you and everyone in the audience, all of our DNA is a little bit different. And that dictates, how we look and our health profiles. So we expect it to be a little different. And sometimes we have changes in genes that are variants that have no effect on the function of the gene. They'd have no effect on the protein product of the gene. But other times we have likely pathogenic or pathogenic variants or changes within genes that affect the function of the gene, and they cause a genetic disorder. So now that we have that background down, I'm just going to move on to some facts. Whenever we think of genetic disorders in Pakistan, the first disorder that comes to mind is thalassemia, because it is the most common single gene disorder in Pakistan. And the second thing that always comes to mind when we think of genetics, we think of consanguinity. So consanguinity is when you are married to a blood relative. You procreate with a blood relative. And when we think of consanguinity, of course, we think of autosomal recessive conditions, and thalassemia is an autosomal recessive condition. So for thalassemia, we know that 8 of 100 Pakistanis are carriers of beta thalassemia. And each year, there are more than 5,000 newly affected births of beta thalassemia. And then there is this study, which is the pie chart that I've made on the side. And that's looking at the individuals who have thalassemia. Are they homozygous for the pathogenic variant, or are they compound heterozygous? Homozygous means that on both alleles, the one that came from mom and the one that came from dad, you have the exact same genetic change or pathogenic variant. And compound heterozygotes are that you have a different variant from the mother and a different variant from the father, but both of them are pathogenic, so they're causing the disease together. But as you can see from this in this chart, the study was looking at whether we are more likely to see compound heterozygotes or homozygotes, and the 83.5% is homozygous. So basically this is just showing us that in parents who are first cousins, they are both carrying a disease allele. They're both unaffected carriers, but they're both carrying the same diseased allele that is then causing the condition. So more common in families that are related. And then some more facts for Pakistan is that the first point that I have there, inborn errors of metabolism, are 10-fold greater in Pakistan than some other countries. And they are an important cause of morbidity and mortality in neonates and children. And this came out of Dr. Homa Chima, which a lot of you might know her. She did some excellent work and she reported this. And she documented how these inborn errors are much higher than other European populations. And also, similarly, there was another paper that looked at neuromuscular disorders, and they were 3-fold higher than in European populations. And the third article that I've listed there, that was a review analysis. It was a meta-analysis. They looked at a whole bunch of papers, and they did a review study. And in that, they saw that 90% of genetic neurological disorders occur in consanguineous families. So this just gives us an idea that there is a lot of genetic disorders, genetic conditions in Pakistan. So it's an important aspect that we need to really sort of increase awareness about and think about some more. So moving on, what are some indications that warrant sort of a genetic investigation? So of course, congenital anomalies, when a baby is born with one or more birth defects, heart defects, cleft lip, cleft palate, any skeletal anomalies, spina bifida, or other neural tube defects, club foot, gastroschisis, really any sort of birth defects. If there's more than one birth defect, then it's more likely that there's a genetic etiology. Also developmental delay or developmental regression, intellectual disability, dysmorphic features, so the facial features or other parts of their body features that are different than what we would expect, failure to thrive, poor growth, and even overgrowth, autism, seizures, and of course, if there's a family history. And this is not a comprehensive list, but it gives us an idea of something that would definitely warrant a genetics investigation. So next, I'm going to talk about some genetic tests that we offer here. And this is the list of the tests that I will discuss, starting with the karyotype. A karyotype is also called chromosome analysis. And it is ordered when we are suspecting an aneuploidy. So an aneuploidy is not the right number of, say, 46 chromosomes, right? So if we think they're extra or missing chromosomes. So a common example is, of course, Down syndrome, Turner, or Klinefelter. And also in cases of ambiguous genitalia, because then that could lead to maybe one of those aneuploidies. It detects extra missing chromosomes, so entire chromosomes, if they're extra missing. It detects large deletions and duplications. And it detects translocations, both balanced and unbalanced. So if one piece of a chromosome is detached and attached to another piece, or if a piece is missing, it'll pick those up, and inversions. So a piece of a chromosome is inverted. And what's important to remember is that the resolution is only about 5 megabases. So if it's a small deletion or duplication, this will miss it. It, like I said, can't detect small deletions, duplications. It can't detect mutations or pathogenic variants within a single gene, and it can't detect low-level mosaicism. So mosaicism is when certain cells have the change while others don't. This is a picture of a karyotype, so it's arranged, it's banded. So you can see the bands, the light and dark bands. So the chromosome, chromatin-rich and poor regions along the chromosomes. And over here, you'll see that there is an extra copy of chromosome 18. So this would give us a diagnosis of trisomy 18. Next, I'm going to talk about the chromosomal microarray. So this is a test that is often first-line testing when there are multiple birth defects, intellectual disability, dysmorphic features, delays, even for autism. Also, when you suspect a specific microdeletion or duplication syndrome. So examples of those, common examples are 22q11.2 deletion syndrome, 5p minus syndrome, or Williams syndrome at 7q. So this can detect copy number variants. It can detect deletions, duplications that are too small to be picked up on the karyotype or the chromosome analysis. It can also pick up regions where there is loss of heterozygosity. So both mom's alleles and dad's alleles on each chromosome, they're looking exactly the same. And that is suggestive of consanguinity. Or another thing called uniparental disomy, when you got both, say, chromosomes from mom instead of one from mother and one from father. It cannot pick up certain things like balanced translocations or inversions and specific point mutations. Also, it can't give you the structure of the chromosome. So this is just looking at losses and gains. So it's looking at missing or extra. It can't give you a structural arrangement because it's not a picture of the chromosomes like the karyotype was. This is an array plot. So if you look at it, the first diagram on top, this is each little dot is a point on the chromosome, is a point of DNA. And what we can see on the top one, normal is all of the black in the middle. That's what we expect it to be. But on the top one, you can see right at chromosome 22, a red bar underneath. So that's showing a deletion. So this is what the CMA spits out. And then if we zoom into that red, that's the second figure below that, where you can see a lot of that red, which is falling under. So that's showing us a deletion. So that's kind of what the lab sees when they're doing a chromosomal microarray. So some other tests that I'm not going to go into a lot of detail is methylation analysis, and that's specifically when you're suspecting a methylation disorder such as Prader-Willi syndrome or Angelman syndrome, Beckwith-Wiedemann or Russell-Silver syndrome. And this shows up with an abnormal methylation pattern. So these are epigenetic changes, not genetic changes themselves. And repeat expansion testing is also a separate test that's done through a different technique, which is Southern blotting. And that you are doing when you're thinking of any genetic disorder that's happening because of a trinucleotide repeat. So the most common one, especially in children, is Fragile X syndrome, which is one of the most common causes of autism in males. Others that we see often in adults is Huntington's disease and myotonic dystrophy. Each of them have a separate trinucleotide repeat that repeats in the sequence. And then lastly, I want to talk about newborn screening before I move on and talk about some interesting cases. But newborn screening is a test, and I live in Houston, in Texas. In Texas, newborn screening is required by law for all newborns. So every baby gets a heel stick. There are 2 newborn screening tests. So one is between 24 and 48 hours after birth, and the second one occurs between 7 and 14 days of age. And they screen for about 57 rare genetic, metabolic, endocrine, hemoglobinopathies and other disorders such as amino acid disorders. It all started with PKUs in the 60s. And then there's maple syrup urine disease, galactosemia, congenital adrenal hyperplasia, cystic fibrosis, fatty acid oxidation disorders, just a whole slew, 57. And it's done because you can identify these very early on, and often you can change diet, you can restrict certain foods, and that helps. So early detection and treatment really helps with the prognosis and outcome. And I see this in clinic all the time. There have been individuals who are adults that were born before a newborn screening. They have PKU, they have severe intellectual disability because of all of the accumulation of tyrosine in their body. And those after newborn screening, where their diet has been controlled and they're perfectly healthy individuals achieving doctorates. So it makes a huge difference. And my hope and my wish is that, inshallah, in the future in Pakistan, we're going to have newborn screening. Next, I want to talk about sequencing. So sequencing is reading through the alphabet of the DNA, right? So DNA is very long, large strings of alphabet, our nucleotides, adenine, cytosine, guanine, and thymine, and it's really hard to read that. So the way DNA sequencing works, this is next generation sequencing, is that they're broken down, they're fragmented into smaller pieces to make it easier to read. We then read those pieces and then they're overlapped. So they find these overlapping to sort of match them up, and then they come up with the final assembled sequence, and that's how we read it. We compare it with the reference sequence to see what the changes are, and then those changes are reported out. So there are several sequencing tests, and I'm going to go through each of them. The first one is single gene sequencing, multi-gene panels, whole exome, and whole genome. So single gene sequencing is when you're looking at a specific gene, and that's when you're really considering a disorder that has a single gene involved. So a good example is cystic fibrosis. Cystic fibrosis is on the newborn screen. They look at immunoreactive trypsinogen levels. If they're high, you screen positive. But again, newborn screening is a screening test. It's not diagnostic. There are lots of false positives in newborn screening. So then we want to do a diagnostic test. So they sequence the cystic fibrosis or the CFTR gene. Another example is Marfan syndrome. If you meet the Ghent criteria, which is the criteria for Marfan syndrome, then to confirm diagnosis, we might sequence the FBN1 gene, NF1 gene for neurofibromatosis type 1. So really high suspicion when we are thinking about a specific genetic disorder that is associated with the single gene. Then you can have multi-gene panels, and the multi-gene panel can even just have 2 genes on it, or they can have hundreds of genes on it. And examples are for, say, osteogenesis imperfecta. We have collagen 1A1, collagen 1A2, and then a slew of recessive genes as well. For seizure disorders, a whole bunch of genes that are implicated in seizures. Cardiomyopathy and arrhythmia, I ordered this panel a lot. So it's a panel that looks at cardiomyopathy genes like hypertrophic cardiomyopathy, restrictive cardiomyopathy, dilated cardiomyopathy, long QT syndrome, a whole bunch of arrhythmia conditions as well. So high suspicion for a specific disorder, but there's genetic heterogeneity, which means lots of genes are involved. Next is whole exome sequencing. This is going back to the exons, right? The coding regions of genes, protein coding regions. So that is what we are sequencing here. This is only 1 to 2% of the genome. So it's a very small proportion of genome, but what we have seen is that it is responsible for 85% of genetic disorders. So it's a very rich sample of exons. And we're going to suspect this when we think there's a genetic disorder, but we don't know the specific disorder. So we provide the lab with the phenotype, the features and the symptoms, and then they sequence all 25,000 genes, the exons of all the genes, but they report back only on any conditions that match the phenotype that we have provided the lab. So it's a phenotype-driven test. So if we don't say seizures, they won't report back on seizures, the related genes. And then we have whole genome sequencing. So this is more near, but we're ordering it much more now. This is coding the entire genome. It's the exons and the introns, the regulatory regions, everything. This is picking up not only problems in the exons, but also if there's an intronic change that's affecting a splice site or the way the intron exons are cut, and so on. It's looking at our mitochondrial DNA. It is also looking for CNVs, copy number variants, those things, the missing and extra that we were doing a CMA for. It's picked up on genome. It's also looking for the triplet repeat disorders that I was talking to you about, like Fragile X syndrome. So this is an all-in-one. So if I wanted to do an exome and a CMA on someone, I would say, let's just do a genome, because it's going to cover the deletions and duplications, and it's going to cover the sequencing as well. Also, if you have a negative exome and all testing so far has been negative, you can consider a genome at that point as well. So these next figures are again going back to Pakistan, and what we have seen in some publications, again, Dr. Chima did the first one, and then the second one is out of AKU, Dr. Kirmani in Karachi. But what these are showing is that genetic testing, when it's done in Pakistani patients and Pakistani children, the diagnostic yield is much higher than even here in the United States. So that's what our first one is saying, that there was a 53% positive genetic testing result and 27% negative for neuromuscular disorders. The second one is showing 61% with exome testing. And over here in America, the diagnostic yield for exome is around 40%. So it's much more than what we would expect here. So we're picking up a lot of pathogenic variants and we are diagnosing a lot of disease in Pakistan. And then the last bar graph, it's just showing us, the diagnostic yield for neuromuscular disorders across different ethnic groups, and Pakistan then again having the highest when compared to European, Latin American, and North American, Australian. So I'm going to talk about a case real quick. So this is a Pakistani family. So this was a case we saw here in Houston, but the family was from Karachi. They initially presented to neurology. It was an 8-year-old male with macrocephaly, failure to thrive, and hypotonia. Neurology ordered metabolic testing and a brain MRI. They referred him to GI for failure to thrive because he had the failure to thrive. So the metabolic testing, plasma amino acids, normal, acyl carnitine, normal, urine organic acids showed excretion of N-acetyl aspartate, and the brain MRI showed demyelination. So they came up with a provisional diagnosis of Canavan disease, and they referred him to genetics. Canavan disease is a leukodystrophy. It's degeneration of the white matter, and you see demyelination along the neurons. It's an autosomal recessive disorder, so we know that both parents are obligate asymptomatic carriers. They can either be homozygous or compound heterozygotes. And they have a 25% chance of having an affected child and a 50% chance of having a carrier. Symptoms, it's a pretty progressive kind of bad prognostic disorder, delayed milestones, low muscle tone, large head size, difficulty tracking objects, reduced sucking ability, blindness, seizures, difficulty swallowing, sleep disturbances, and there's a shortened life expectancy, death around 10 years old. Some live into their teens. Treatment is really just symptom-based with anti-epileptics and a G-tube, physical therapy. So we tested the patient. When they came to genetics, we sent out genetic testing. It was a single gene panel because we were pretty sure of the diagnosis of Canavan disease. They really fit based on brain MRI and labs. And it came back positive for Canavan disease. So the ASPA gene is what causes it. And the variant you can see over here, it's described. Heterozygosity, it was homozygous. We'd expect him to be homozygous because it's a recessive condition and it's pathogenic. They classify that variant as causing the gene to not function. Then we tested mother and father, and both of them, as expected, were carriers for the variant that the child was homozygous for. So they were both heterozygote carriers. So if we look at this pedigree over here, this is just kind of talking about how consanguinity increases the likelihood of autosomal recessive conditions. So if you look over here, the red little a is a rare recessive allele. It's a rare recessive allele. It occurs sometimes, but you need 2 little a's or to be homozygous to actually have the condition. In generation 2, both the children inherited the little a from their father, the pathogenic allele from their father. But again, they're asymptomatic because they're just carriers, because this is a recessive condition. In generation 3, their children inherited the pathogenic allele from their parents, but they're first cousins, and they got married and they had a child. So now that child inherited the rare recessive allele from both of their parents. So consanguinity increases the chances of someone to have an autosomal recessive condition because they have an ancestor in common that has or carries a rare recessive allele. And that's what we see over here. And I've just calculated these probabilities for you. But just in this case, because cousins were married, there's about a 6.25% chance that the child is affected. And if this individual, the one that the arrow is coming down from, married someone in the general population, if we look at the incidence of Canavan disease and calculate the carrier frequency in the general population, it's about 1 in 150. So that the probability was about less than 1%, right? Much less than 1%, 0.01%. So consanguinity is like very culturally accepted common practice in Pakistan. And there are a whole bunch of reasons for that. And in one of the papers I published, I had described all of these reasons. So it's not my place as a genetic counselor to tell anyone not who to marry or not to marry. There are people in the audience, of course, that are married to our cousin. There are people in my family that are married to a cousin. So that's not what we're doing. But what we are doing is providing them with information so they make informed decisions. And that's what happened here. So this is the pedigree of this family that we saw, right? And their first cousin. So when we were looking at this pedigree, I looked at the folks on dad's side and mom's side. And I said, any of your sisters getting married to any of the brothers on that side? And they said, yes, abhi Pakistan ja rahe hain December mein mangni ke liye. So I was like, oh, okay. So I said, well, should we see if you're a carrier? So we tested dad's sister and she turned out to be a carrier. So then we're like, okay, so you're a carrier. So you're going to Pakistan and you're, having your mangni with one of mother's brothers. So now we need to see if he's a carrier so we can determine what the risk is for your child. So she was going in December, I gave her a kit and you just had to get like a sample from the mouth and bring it back. And then she came back in January, called her, saying, did you bring the sample? She's like, no, I didn't. Like, oh, no, you didn't bring the sample. We can't do the testing. We won't know if they're a carrier. And she's like, well, no, our families talked about it and we decided to break the engagement and not get married. And I was like, oh, really? She's like, yeah, no, it was fine. We just discussed it because, this child, she's living with her brother in the US and she sees this child who is going to have an early death, severely affected. So they made that informed decision. So really my role as a genetic counselor is to provide individuals with the information so they know what to expect, they know the prognosis, they know the recurrence risks, and then they can make their decision. And that's what's written on this slide. Next, really quickly, my second case is very interesting, but this was a 6-year-old who was referred by their pediatrician to genetics for bilateral cleft lip, cleft palate, hypothyroidism, oligodontia, missing teeth, delayed milestones, walked at 2, and words at 3. When he came to the genetics clinic, we do, the geneticist does a physical exam. He had a tall forehead with a high anterior hairline. You can see in the photo there. He had cutis aplasia, so he was missing a part of his scalp that had healed when we had seen him, but you can see where the hair is missing. Sparse and coarse hair, sparse eyebrows, ectropion of the lower lid, so they were tilted outwards like in the picture over there. Distichiasis, which is an extra layer of eyelashes. So there's one layer of eyelashes and then a whole next layer of eyelashes. Epicanthal folds in the inner corner of the eyes, a prominent beaked nose. You can see that in the picture. Repaired bilateral cleft lip, cleft palate. He had few conical shaped teeth, a simplified right ear, and multiple hyperpigmented scars on his legs, which you can see there, and hyperplastic nails. So genetic testing. We did a CMA looking for microdeletions and duplications in 2013, negative. We did exome sequencing in 2014, unrevealing, it didn't show anything. We did a reanalysis. With exome sequencing, you can ask the lab to reanalyze the sample because there are new genes, they have new algorithms, and they can see if we find something. So in 2016, we didn't find anything. Then in 2018, the West came back positive, and we found a change in the CDH1 gene that gave him a diagnosis of this condition called blepharokelodontic syndrome. And the reason why it came back in 2018 positive is because this article was published in 2017, so they had new information to go by, in which they were saying that there is this condition, blepharokelodontic, which affects the eyelids, the upper lip, and the teeth. And they did whole exome sequencing in 11 individuals, and they found pathogenic variants in 5 of them in the CDH1 gene. And it's autosomal dominant inheritance, and there's variable expressivity because all of these different people presented with different symptoms. The CDH1 gene makes E-cadherin, which is a protein that's essential for normal development of the eyelid, the tooth, the hair. So it makes a lot of sense. But what we also know about this gene from before is that CDH1 is associated with hereditary diffuse gastric cancer and lobular breast cancer. However, this article that was published, they said that none of their patients, none of the 11 had any cancer. So we're thinking that, you can have a phenotype that's just this ectodermal dysplasia phenotype and not cancer. And that's what we think it is. This is just a list of some of the features, and all of the ones that are in red are the ones that our patient had. So he really fit down to the hypothyroidism, which goes with it as well. Then in 2020, 3 years later, there was another paper that said that no, it's not that it's a separate phenotype. We report a patient with blepharokelodontic syndrome that also has diffuse gastric cancer. We see the patient back in clinic and we say, in addition to everything else you're doing, you also need to have screening for gastric cancer. So that really changed, right? So this went from him not having a diagnosis for so many years to him having a diagnosis. And of this ectodermal dysplasia, the blepharocheilodontic syndrome, and then him also having a secondary diagnosis of him being at an increased risk for gastric cancer. So the benefits of a genetic diagnosis: medical, anticipatory guidance, targeted treatment, management, surveillance, prevention of complications, and improved prognosis. Emotional and psychological validation that this was not caused by the parents, relief from the uncertainty of being in this diagnostic odyssey, reduction in stigma, improved mental health. Family and reproductive informed marriage and reproductive decisions, like in my first case, identifying at-risk family members, and avoiding misdiagnosis in relatives. So lastly, I just want to say that I am one of the founding members of the Pakistani Society of Medical Genetics and Genomics here in the United States. And I have other geneticists and genetic counselors that are part of this organization. And we are really trying to enable provision of affordable medical genetic services to the people of Pakistan, to create awareness of genetic disorders through education and research and community outreach. And Dr. Sadiq and Dr. Safdar, they've all been at our meetings, and they're all involved in this effort with us. So they are definitely our partners. And I haven't talked a lot about what's available in Pakistan, but here are two publications that I've authored. And these two publications talk about sort of what's available in Pakistan in terms of genetics and what the needs are of the country. And we're really hoping that Dr. Sadiq is going to fulfill some of those needs. So thank you for listening. I hope I didn't go over time. And again, thank you for inviting me to speak.
Hello. Thank you, Dr. Maila Ashfaq, for highlighting a very important topic of genetic decoding and knowing about the future of the children. Of course, it looks very, very fascinating. And in the recent past, I think we started this genetic testing, right? We started from Lahore, from Professor Humash, under the guidance of Dr. Arnold Rao. And then it was disseminated throughout the country in Islamabad, Karachi, Multan. Of course, it looks very, very fascinating. But as you all know, the poverty in Pakistan is touching around about 42%. And there is a lot of expense which is done on this testing of genetic whole exome sequencing and genetic testing, which is minimum is about 150,000. And if that doesn't give you any idea, then you have to go for the genomic testing. And I just see the syndrome which you describe, our blepharocheilodontic syndrome, which was diagnosed after 4 testing. So in a country like Pakistan, we can't afford to go for the 4 testing. And even if you have got some sort of a diagnosis, you are absolutely right. We can guide, not in terms how to manage these children, and most of these genetic disorders are not curable. But you're absolutely right. We can anticipate the complications which are developing over a period of time. We can take care of those complications. And then guiding the parents about the next pregnancy. Again, you had to pay the money, because if you had to go for the chorionic villus biopsy, again you had to pay a genetic testing. And the parent immediately, doctor, if the child, next generation is again having the same problem, what are we going to do? So I think these are the very, very difficult questions. And in terms of money which has been spent on these genetic disorders, so these are certain queries in our mind being a Pakistani. So what is your advice about these?
No, you're absolutely right. But dekhein, 10 years ago, 15 years ago, there was no genetic testing, right? It's not like Pakistan can stay in the dark ages. Pakistan has to get to where other countries in South Asia are. China, India, all of these places have genetic counseling training programs. They have genetic testing in-house. Hopefully, that's where we're going to be. I understand that it's costly. However, the price is decreasing substantially. Again, 12 years ago, an exome was started off at $12,000. Now it's under $1,000, right? So the price is going to continue to decrease with advances in sequencing technology and so on. And like you said yourself, making a diagnosis is important for many reasons, right? And I listed those reasons. And while we're paying attention to communicable diseases and we're looking at malaria, we're looking at everything, definitely, we have to do all of that. But we can't be lagging behind in genetics. We need to be moving forward. And I feel like right now we have moved forward. Just in the time since we started PSMG, so much more has been done. 10 years ago, my first publication was when I was at Stanford. And if I compare to what's happening now in Pakistan to what was happening then, leaps and bounds. So I just feel like we'll just continue to move forward. And eventually, hopefully, there'll be programs in place, newborn screening, Punjab government do something, do stuff for the people, and make these diagnoses. And there's a lot of treatment. Enzyme replacement treatment for newborn for metabolic conditions is happening. Now we're going into CRISPR, and that's a whole other talk. But there are a lot of treatments that I think will be available. So we have to, just kind of keep moving forward. You made excellent points, Doctor, but we just have to keep moving forward. We can't let Pakistan stay behind.
Thank you very much, Myla. That was a very comprehensive closure, I would say. We have two more chairs sitting with us, so I would be requesting both of them to have few comments before we move on to the next plenary talk. And Dr. Safdar, if you wanted to say something, being President APPNA Pediatrics, you are welcome, but I would let both my colleagues say a few words before we move on to you. And then I think it will be up to you to stay. We know that it's middle of the night in US, and we are extremely grateful for your contribution and being part of this plenary session at this time. Over to Dr. Muhammad Hussain and Professor Naeem Zafar, and then to Dr. Safdar before we move on to our next plenary talk, because that's in-house. Professor Rizwana is going to do that talk. Over to Professor Muhammad Hussain.
Thank you very much, APPNA, and we appreciate your dedication at this late hours at US. We have discussed three key areas about the screen time, AMR, and genetic disorders, all central to the health of our children. I'm extremely thankful to all the speakers, but just a question for the Dr. Zishan, that does the excessive screen time have a measurable impact on intelligence, attitude, and memory, or the primary concerns are limited only to the vision and the issues of the neck and the back muscles? And the second question is greater harm attributed to the duration of the screen time or the screen use, or it's the nature of the content that we consume? Thank you.
Thank you so much for asking the question. So the greater harm because of the content you ask, and the first question, sorry, if you can repeat this first question.
Sorry, my first question was, does excessive screen time have measurable impact on intelligence, attention, and memory of a child, or the primary concerns are limited to the vision or the musculoskeletal problem like the neck and back muscles due to the extreme time of the...
No, thank you, and apologize for asking. I know it's the middle of the night, and you're, in Pakistan, you get the really good chai. We are missing those things. So please apologize for my memory. It's not because of the social media, it's just because of the middle of the night. So to answer your first question, yes, I think there are significant, I can share in my private practice. So during my training at Boston Children's Hospital, we have a clinic, which was at that time the first clinic in US where we are seeing children who are struggling with the screen addiction and the screen overuse. And I cannot tell you how many times we have this difficulty. They are affecting with the sleep, irritability, decreased school performance, poor frustration tolerance. And as long as their attention span was significantly, I don't know about the IQ, but definitely their attention span, their memory, all those things were significantly affected, both excessive overuse, the second, lack of sleep, the third, that they are in this virtual bubble, but when they go in the real world, they were struggling to make those connections, they were struggling to, have the patience. So to briefly answer your question, I don't know about the IQ, but definitely memory and attention we have, there are impact on it to the point that, the school performance, the relationship, all those things have significantly getting affected. If you talk about the time or the content, I think both are matter, but definitely if I have to pick one, definitely the time. If the kid is using 4 hours, 6 hours of screen time where it is affecting their sleep, where they are not able to pay attention in their work, where they are missing out their social connection. And I don't know about in Pakistan, but in US, there are so many cases now. There are virtual, friends, and where they are chatting with each other. There are a lot of child predators, people who are asking for money, people who are asking. All these things are happening the more you use. As far as the content, definitely, especially for teenagers, anything that has more visual, like an Instagram, definitely has more video, more visual content. TikTok has more impact. Similarly, the content which has more violence, so it has definitely impacting their behavior as well. But if I have to pick, I will go with the time of the social media.
Thank you very much, Dr. Zishan. That was very comprehensive response. Over to Professor Naeem Zafar before we move on to Dr. Safdar Sultan.
Thank you very much, Professor Masood Sadiq. In fact, Dr. Zishan and Dr. Mubin Rathore are both dear friends. Zishan being the youngest executive board member of APPNA, a frequent traveler to Pakistan and works diligently on infant and child psychiatry, working closely with Pakistan Pediatric Association, especially the Child Rights Group. And his work spans not only in the field of psychiatry or psychology, but also all aspects of mental health and child health. And I'm so grateful for all your talk on the screen time. Just mentioning that we probably missed out on just one very important area, which is the online violence against children. And obviously that was another area that we would have thought you might would like to tell us about. Mubin Rathore being a very dear friend and my classmate, also a frequent visitor and a stalwart, and he fights for prevention of infections. And I must say, what he preaches is prevent it before it happens, and the best intervention is hand washing for 20 seconds. That's the key messages that he gives everywhere. Talking about Myla, I totally agree with you, Myla. When I started working on child abuse and neglect and on mental health and on different aspects of developmental pediatrics, we were faced with the dilemma of survival of children. People were dying of diarrhea, dying of malnutrition, and everybody said, why do you talk about abuse? Almost 30 years down the line, now we know the importance of these subjects as well, and we are talking about them. So definitely genetic counseling, which was not available at that time, it is now available. It is going to be useful, and saying that we cannot afford genetic counseling right now is not the right thing. Everything has to go into the right perspective, and hopefully in the next few years, the prices will also come down, but the cost, it is not just the cost, it is the importance of that which is going to rise substantially. Thank you very much.
Thank you very much, Professor Naeem Zafar. Over to Dr. Safdar Sultan, President APPNA Pediatrics, for closing remarks from US, please.
Assalamu alaikum. First of all, thank you so much. I would start with thanking the leadership of PPA and the organizing committee in Quetta. You are all there in person, which is great. And I think and then I would like to thank our speakers. They were all three excellent talks, Zishan, Myla, and Dr. Rathore. Thank you so much. And I think this was our first hybrid experience and first session. I think it went really well. We should continue this activity, and I'm a strong believer of subconscious learning. I think when you interact and collaborate and you have these sessions, they make a huge difference in the long run. I think the more we do, the more knowledge sharing we do. Honestly, I was listening to Zishan and Myla and Dr. Rathore, and I learned a lot of new things today, which I'm really grateful. And also, I think we have a generational gap, institutional gaps in Pakistan. We can fill those gaps slowly. I mean, I know there is no magic bullet. We can fix one day, one lecture, one conference cannot fix everything. But I think, for example, as Myla mentioned, we have to start somewhere. And we have to be progressive. We cannot think, we don't have these facilities in Pakistan, so let's not even talk about it. once we talk about it, someone will come with strategy, better ideas to even start doing it. An example is like, genetic testing. At this point, they are expensive. They can become cheaper over time. So, but we should start having infrastructure we can handle when we are ready. So all these things, they matter. And then I don't want to take more time, but I will like to stay for the next session as well. I will stay. It's a weekend, and it's a Sunday next day, so it should be fine with me. But thank you so much once again, and this is a wonderful activity, and I'm so happy I joined.